So, good afternoon, everybody. Welcome to this one-day international webinar on immunological aspects.
সুনির্মলা কথা কেটে যাচ্ছে।
Webinar, when the world as well as India has been altering for most 2 years, suffering from corona disease, that is COVID-19. And at first, I like to inform you that our Honorable Principal Sir and Chief Patron of this webinar cannot join this program due to
কি প্রেজেন্টেশন আবার কে করে দিল?
সুনির্মলা আনমিউট করো।
সুনির্মলা শোনা যাচ্ছে না।
Shamita,
হ্যাঁ,
Madhusudan, how are you audible?
Yes.
Yeah. So, welcome everybody. Again, I try to welcome you, all the participants, to this international webinar, which is organized by the Department of Zoology, Belda College, the entitled Immunological Aspects of COVID-19. I request all the participants, please mute your sound and video to avoid background noises that may distract you and others from listening to the webinar. And in the absence of Principal Sir or inaugurator of this program, Dr. Manavendra Mondal, Chief Patron of this webinar, I invite as well as request Dr. Madhusudan Diwari, Assistant Professor of Department of Zoology, Belda College, and Secretary of this webinar to deliver our inaugural address of the webinar, the international webinar. So welcome Dr. Madhusudan Diwari, please.
Thank you. We are audible?
Yes.
Good afternoon, everybody. Infectious coronavirus disease is caused by SARS-CoV-2 virus. Most people infected with the virus will experience mild to moderate respiratory problem and recover without requiring special treatment. However, some will become seriously ill and require medical attention and may die irrespective of age. The best way to prevent and slow down the transmission is to be well informed about the disease and how the virus spread. Till date, there is no specific medicine recommended to prevent or treat the coronavirus. A strong immune system helps to protect the body against virus. Therefore, it's quite essential to build natural immunity. It's the only way to prevent coronavirus disease and be healthy. In this context, for our healthy life and also for our survival, it is great pleasure to us that Zoology Department of Belda College have organized a webinar on immunological aspect of COVID-19. I am thankful to our Honorable Principal Sir, Dr. Manobendra Mondol, for his encouragement, continuous guidance, and support. I give my heartfelt thanks to Dr. Shobhan Rai, scientist, Department of Biochemistry and Molecular Biology, University of Chicago, USA, and Professor Pradeep Kumar Das Mahapatro, Department of Microbiology, Raiganj University, for their august presence in this webinar as our valuable speaker. Lastly, I thank to all participants and my colleagues for their cooperation to make this webinar as a grand success. Thank you, thank you.
Thank you, Madhuchanda, Dr. Madhuchanda Tiwari. Now I invite Shomita Maiti to introduce our first speaker, Dr. Shobhan Rai. Shomita.
Good afternoon, everybody, and thank you, Sunil Malda. It is a great pleasure for me to introduce our respected speaker, Dr. Shobhan Roy, who is going to talk about immune response to COVID-19. He is a staff scientist of University of Chicago. He is a protein biochemist and structural biologist. He is a protein biochemist and structural biologist working on the molecular aspect of the human immune system in University of Chicago. He has completed his PhD from IIT Kharagpur in the field of biotechnology. He was also postdoctoral fellow in the University of Chicago. He also got Eli Lilly Asia Outstanding Thesis Award. He has published 14 research papers in different reputed international journals like Science, Science Immunology, Proceeding of National Academy of Science, and the Journal of Immunology, Cellular and Molecular Immunology, Virology, Journal of Structural Biology. So on behalf of our department as well as webinar organizing committee, I welcome our respected speaker, Dr. Shobhan Roy.
Thank you, Sunil Malda. Thank you, Madhuchanda. Thank you, Shomita. Thank you, Professor Das Mahapatro, Pradeep. So it's a great pleasure for me also to connect with Vidyasagar University, and I'm very happy to see that I'm like already I know many people which I have been and not talking about for many years, but I can see here. And আরেকটা কথা বলতে চাই প্রথমে কালকে তো মহালয়া সবাইকে শারদের প্রীতি ও শুভেচ্ছা জানাচ্ছি ভালো করে সবাই পুজো কাটান খুব আনন্দ করুন so now I'll share my presentation So can you see my slide?
Yes.
Yes. So here, আমরা পড়েছি ভূগোলে যে হ্রদ অঞ্চল there's like 5 different lakes in US. So the Lake Michigan, which is and Chicago City actually is very close to the Lake Michigan, and University of Chicago is more than a century old. And it's a huge campus. You can see these are not entirely, but my field of research is human immune system. And as Sumita was saying, generally by training, I'm a protein biochemist and structural biologist. So I like to understand at the immune response at the molecular level. And you can see this the pictures, which is showing this round shape, and which is actually coronavirus CoV-2. And the name of this virus actually came from, because you see this is like a crown or the mukutan moto. So that's how the name actually came. And this is actually electron micrograph of this coronavirus emerging from the cell. So this is the brief overview of the talk or the different aspect I would like to touch, like very brief introduction about immune system, how does our immune system work, and what are the different components of it. And then I would like to talk about like why research is important, and like especially the structural biology or the biochemistry is why these are important aspects, and how they help in vaccine development or understanding the like the pathogenic infection or immune system. Then I'd like to talk about coronavirus, and then I'll go to the human immune response towards human coronavirus. And then I'll talk about two specific cells, like those are present in our body, is B cells and T cells, and how they actually work in immune response against coronavirus. Then I'll talk about the vaccine development, different strategies, and finally I'm going to the conclusion. And hopefully I'll be able to show. So our immune system made up of like many different kind of cells type, and broadly they are classified into two different categories, like innate immunity and adaptive immunity. Innate immunity is a very rapid response, like something bacteria or virus when enters into the our body. The innate immune cells like basophil, eosinophil, neutrophil, all these cells, macrophage, dendritic or mast cells, they actually act very rapidly. And they basically recognize some part of this bacteria, which is called a pathogen-associated pattern. So they will recognize those and act very quickly. Whereas, like the other part of the immune system, which is adaptive immune system, they're slow. Like the B cell and T cells, they usually take time to mount an immune response against pathogen. But they're different from the innate immune response because they have two different characteristics. Like they are very specific. They can be they can specifically recognize for one pathogen or virus or something, and they can have memory. So suppose there was one infection with some virus or something, and these B cells actually remember that, or T cells remember that. So basically, our body is remembering the past infection, and so when the infection again happen in the second time or third time, they will act much faster. So that's the key for the vaccine development. Like what we're trying to do, we are training our immune system so that they can recognize those pathogens and they can act actually at the time of infection. And B cell and T cells, they have specific receptor for to recognize these pathogens. And as you know, like we always talk about antibody. So antibody is kind of a Y-step molecule, and which is also the same like B cell surface receptor or BCR. And it has this binding sites here, like which is the heavy chain and light chain. They actually prepare this binding site, and it can bind to anything. That's the beauty of antibody. And also, like antibody is actually secreted from the cell. So maybe the B cells is sitting somewhere in the body, but they are secreting into the blood, so they can go and like bind a pathogenic bacteria or virus from a different and distant place. And the T cell receptors is very different. T cells is receptor or T cell is always bound with the plasma membrane, and it's a dimer of like two proteins. It also has like only one binding site, like here. But whereas the antibody has like two binding sites, and it can bind like two different antigens same time, but they will bind to the same epitope at the side. Another important aspect of the TCR is like it will not bind antigen by itself, so it needs some help from other molecules. I'll talk about that in the later, but antibody can bind to an antigen by itself. So this is just to show like one of this how antibody how does antibody looks like. This is one of the crystal structure of the antibody. So so that's why, like, I am a structural biologist, and I like to see how does this molecule look, so that we can understand at the molecular level, and also we can manipulate its function, so that can be used for drug or some other purposes. So antibody can be used as a drug. Like there are a lot of uh, like antibodies already available in the market for the treatment for cancer or some other. So, and these two place is called the antigenic binding, where the antigen actually binds. And as I was telling you that antibody can bind anything. It can bind whole virus. Like you can see one of the like picture of a virus. It can bind to the virus. It can bind to bacteria, or it can bind to proteins, and it can bind to the sugar. But specifically, like if you think about virus, like basic or the bacteria, they are made of a structure which is actually composed of proteins or sugar. So basically, antibody is targeting those virus or the bacteria through binding with the protein or sugar. But again, most of the virus, they have this like this outer membrane protein or spike protein. Those are also like highly glycosylated. Like they are attached to its sugar. So you can see here, like all these, like not the white color, like the red or the blue. But these are basically sugar molecules. Those are attached to the protein. So then that will make difficult for antibody to bind this protein. So that's the strategy for virus to evade the immune response as well. So this is the crystal structure of the crystal structure of the TCR, so that you can we can understand the difference between the TCR and the antibody. So you can see it's just a dimer of the two protein, and they call, like, sometimes they call alpha chain or beta chain based on their sequence. And they can't bind antigen by itself. So they need help from some another specialized molecule is called MHC, major histocompatibility complex. And this is class 1 MHC molecule, and they are also very specialized. What they do, they actually bind like a small part of the peptide or antigen, which is coming from E. coli or virus. And after binding, they will present those into the cell surface, so the T cell receptor can come and bind to this antigen, and that's how our immune response actually starts. So you need always T cell receptor to recognize one of those ligand, and then T cells can actually modulate the immune response. So this is one of the simplistic view, like how this antigen processing and the immune response starts. Like anything foreign, alter, or tumor antigens, when they are in our body, we have some kind of specialized cells, which is called antigen presenting cells, like macrophage, dendritic cells, B cells. So what they do, they usually take up those like bacteria or virus. They'll eat those, and then basically they will try to like digest those and make like small species of antigen, and those will bind to the MHC molecules and present on the cell surface. And the T cells, which are everywhere in our body, they are always scanning for something which is not our cell, like something not our, or is coming from actually from the environment or the foreign. So they will recognize those very specifically and start the immune response. Basically, they will produce cytokines, so other cells will be there, and they will start fighting with the disease. So how does the immune system distinguish between self and non-self? So like I was telling, the T cells are the important in terms of distinguish between which is self, which is not self. So basically T cells, like we need to know the specific structure of the T cell receptor. So as I was telling you that there are two chains, alpha or the beta. And again, if you look for the alpha sequence, there are three regions in this protein. This is called cluster determinant region 1, 2, 3, and beta also same thing, like CDR 1, 2, 3, and those are actually here, and they come all together and sit on the. This is the MSC molecule, and ELO is the peptide which is coming from a bacteria. So they will come there and they will just try to bind with this peptide and also the MSC at the same time. So they will only bind if this is coming from a pathogen, but they will not bind to any of our own peptide, because in that case we'll have autoimmune diseases. And here you can see that CDR 1, 2, 3 for the beta chain, and also for the alpha CDR 1, 2, 3. So CDR 1 and 2, they are actually binding with the MSC molecule, but whereas the CDR 3 is actually binding with the peptide. So that's how tissue or our body actually recognize which is self and which is not self. And yeah, so that's and we understand, like we able to do that by like structural method. Like otherwise, it will be very difficult to know exactly how does this different part of the tissue is actually interacting with which part of the molecule, and how they're recognizing like which is a foreign particle or the foreign bacteria. So for the structural biologists, they use like X-ray crystallography. So basically, you purify, make those protein, and then purify those and make beautiful crystals if you are lucky. And then if you're lucky, you'll be able to get the data, and then finally you'll be able to solve the structure, and you'll be able to analyze the structure at the molecular level to see like which is the amino acids are interacting with which protein, and why they're important. So now another new technique is also available, which is called cryo-electron microscopy, which is like another groundbreaking technology now is now available. In that case, you don't have to make or protein, or you don't need to have the crystal. Basically, you can take the picture of the your protein or the complex, and then you can solve the structure. So you can see this is one of the cryo-electron microscopy of the coronavirus, and you can see those spike protein, which is actually on the envelope of the virus. So what we are getting, what are the information we're getting from the structurology we are understanding at the molecular level, like which amino acid is making contact with which peptide, and how they're arranged in a three-dimensional space. So all this information is only available through, like if you're able to see those. And these are very important for the drug and vaccine development. So I'll talk about one of the study which we did a few years ago. So the T-cell receptors, also the tissue, also can recognize the cancer antigen, like which is actually not exactly non-self, and it's not coming from outside. It is actually coming from our body, but in normal cases they are not there. So they will only express when only present when there is any cancerous cells in the body. So what we did here, like this is one of the T cells, and we found those T cells in the body, and then we found that okay, they can actually have anti-cancer effects. So we wanted to understand like how these T cells recognizing this particular cancer antigen, which is called MART1, which is a peptide. And this protein, the MART1 protein, is actually expressed when there is melanoma cancer. And so if we zoom in the structure, you can see this is the MSC molecule, and the inside, this is the peptide, which is the MART1. And this is one of the tissue. They come very close to the peptide and the MSC molecule, and these are all the serial loops. They basically recognizing this MART1 peptide. And if we have a different peptide, they will not bind in the same manner, or their affinity will be so low, they will not bind. So that's how, like, this is the technique I usually use to understand the molecular details of the recognition by T cell. So now I will switch my gear here, and I'll talk about coronavirus. Um, so in geology, we had this subject taxonomy and evolution. So this is just to show one of those phylogenetic analysis of the coronavirus, and they are from Coronaviridae family, and there are 4 subfamily: alpha, beta, gamma, and delta, of which the SARS-CoV-2, MERS, all are from the beta coronavirus. But there are, like, maybe we don't know how many coronaviruses are there, but these are the few we can get the sequence, and then we can analyze the sequence and then make phylogenetic tree. This is the left side, right side figure is basically the same, but this is also showing you they are very closely related strain or the pieces of the virus, but they are from different organisms. Like some of them from the bat, some are for the pig, some are for the bovine. So these are the different coronavirus available in the nature, and their structure is very similar. Like they have a positive strand RNA genome. So instead of the DNA, they have RNA, and outer they are encapsulated with a membrane, and then they have this outer protein which is called spike protein. And spike protein is very important because this is the receptor. This will bind to another protein which expressed in the lung epithelial cell, and that's how they can enter into the cell. And the left side figure, this is showing their genomic organization. Basically, you can see that they have a very similar genomic organism. They have this S, which is spike protein, and they have all the different accessories protein here. But also, you see there are also they can be different too. Like they have different composition of the different small accessories protein as well. So based on this, like there are different protein, and people think like they actually can modulate the severity of the infection. So otherwise, they have very similar genomic structure or the actual structure of the virus. So some viruses can be like they can infect others. Like most of the cases, this coronavirus actually present in bat. Bat has they have a different kind of immune system, and they can maintain a lot of virus. So bats are actually host for many different kind of viruses, and bats are fine because they don't get sick with this kind of virus infection, but they can serve as reservoir. So this virus can transmit to the other organism, like in case like the MERS virus, it can go to the camel, and then those viruses will adapt in camel, and then finally it can infect a human, and then that's how it can spread. And SARS-CoV-2 also this is a controversy still, but I think in case of the SARS-CoV, it has been found that from bat it go to the civet, and then finally it came to human, and then it also started transmitting to the human. So there are other coronavirus also, like HTU, OC. These are the different strain of the coronavirus, but they different type of coronavirus, but they are probably mild, so they don't do severe infection most of the cases. But sometimes, like we don't have, because these are the new viruses, and we don't have the immunity that we need for those, so that can create pandemic. And here, just to show like different coronavirus and their natural host. Like most of the cases, this is bat, and sometimes it can be mice also. And so basically, they will jump from bat to some other organism, maybe the camelid or bovine or cipit or camel or pig, and they will add up there, and so virus will change a little bit their protein and their structure, and so they can also infect other organisms. Um, so whenever we are dealing with wild animal, so you have to be very careful uh, because they might have some pathogenic, and those are we are not prepared for those. This is showing the general uh, scheme of a virus. Like when they enter into the cell, they basically try to hijack the cellular machinery and multiply. So here, showing the human coronavirus, which can the spike protein, which binds with one of the human and protein which is present in the surface, which is called angiotensin converting enzyme 2 S2, and then it's entered into the cell. And the virus has this RNA which is released into the cells, and also they carry another protein which is called polymerase, uh, which can copy RNA to RNA. So it will produce more RNA, and those RNA will translate into different proteins. Those are required for the virus, and finally the RNA and the protein they will uh, assembly into a mature virion particle and then finally released into the media. So this is the general scheme for every virus. They just want to enter into the cell, and they try to utilize all the resources into the cells, and then they'll go out. So basically, they'll just keep multiplying and infecting, and this is the cycle usually go. But this is not good for the host. So basically, this will kill the host. So in order to stop this, we have these different kind of immune cells like B cell, T cells I was talking about. So they'll come there and try to uh, mm, build an immune response against these viruses. Like here, this is one of these lung alveolar, and this is the lung epithelial cells. So when the SARS-CoV-2 enters, um, and it can infect the epithelial cell, and it will try to multiply. But at the same time, there are other immune cells like a macrophage or T cells. They will sense that there is some problem, and they will come here and try to produce all the cytokines. In normal cases, this is fine because that will eliminate the virus finally. But in some cases, it will not. So if the immune system is not working properly, so the lung alveolar cells will die, and there will be a Hall membrane. That's basically stop the passing of oxygen from lungs to the blood. So that's how people will have like problem with the breathing, and that's why like people are using ventilation because initially there was no drug available, and people usually don't know how to deal with these patients who are having this serious problem, respiratory problem. So the ventilation was helpful for some time, and then like different and people tried different antiviral compounds. People started doing this antibody therapy and plasma therapy, which is like taking the plasma from actually a infected um, patient, which is clear the disease. So basically take the plasma from there, so it will have this different kind of antibody, and then inject into the patient which is having the serious illness. And second, also vaccine. So vaccine is very important because it can protect us. Like we are prepared before the actual infection happens. So, but the vaccine development is not very easy. It it takes like decades of time to develop one vaccine. Um, so the right hand figure just to show like what are the different like strategies we can take to block the virus infection, like in case of COVID-2. So I was telling that S2 is the receptor for this the spike protein. So we can have antibodies like which can block this interaction. So instead of the binding to the ACE2 protein, antibody can bind to the spike protein, and that block the uh, binding with this protein, so the virus will not enter into the cell. And also we can have like a different kind of inhibitor, like these proteolysis. Most of the cases, the virus make polyprotein, and they have a protease which actually cleave those protein into the different small proteins. So we can have proteolytic like protease inhibitor that can block this kind of function of this, and so the virus will not mature. Also, the RNA-dependent RNA polymerase, which is required for their copying the genome. So it will make more copies of RNA from the RNA genome. So that kind of inhibitor are also helpful. And that time also we were talking about chloroquine, which is found out later that chloroquine is actually not very helpful. So these are the different strategies we can take for the actual, like when the disease is actually happening to mitigate this serious illness. But I think vaccine is very important, and so that because we can vaccinate people beforehand, and that can help people to clear the infection by its own, and they don't have to go to hospital or anywhere. So so what happened, like when like virus enters into the cells, basically cells also have their own way to react. So what they do, they will try to suicide, because by that way they'll stop the multiplication of the virus. So this is another strategy of our body or the cell to not to promote the virus infection. So this is when this happens, and they also release different material like viral RNA, ATP, like host DNA, everything they will release into the media. So these are kind of this will also like go into the nearby cells and alert them, basically something wrong is happening here. And also in the same time, that will bring like all the different immune cells into the place, like T cells, monocyte, macrophages. They will all rush to this place and try to figure out what is happening here. And in the process, they will also produce different kind of cytokine, and that's how the immune response works. And in normal cases, like what happened, like when T cells will come, and T cell will recognize this antigen presented by the cells, and they will eliminate those infected cells. The antibody is there, so basically antibody can bind to spike protein and kind of coat them, so they will not be able to bind with the ACE2 receptor, and they will not enter into the cell. So this is another scenario, and some of them, it's called like neutralizing antibody, which is very important, because many antibody can bind to the like other part of the spike protein, but they may not block the actual interaction with the ACE2. But some of them are actually special antibody, which are we need those actually, so they can block the virus infection. And macrophages, they will come there and they'll kind of eat those virus and destroy the virus. So this is the normal immune response, and then that will cure the infection, and there will be no severe illness. But sometimes what happens when immune system can't work properly, so what will happen, they'll start producing more and more this kind of cytokine, but the virus infection also still keep going to the other like nearby cells. So what will happen this time, like this is basically they'll try to produce a lot more cytokine and everything, so that will have some kind of effect on the other organ too, like that will damage the it's called organ damage or multi-organ failure. So that will come from this kind of like not efficient immune response. So that's why like our immune system needs to be working in a proper way, so that we can so that the cell can recognize those virus and then clear up the infection. So this is kind of immune response part. But now I would like to focus on like how we can develop a vaccine and what is the principle behind it. Like in normal cases, like when any like SARS-CoV-2 or other MTB or any other bacteria when they enter, in many cases, like what happened, like the innate immune response I was talking about that, like they are the fast to response and they are very rapid. So they'll respond immediately, and basically they will eat those pathogen and then display those pathogen and also produce a lot of cytokine. So then other part of the immune systems will be there. So innate immune response, and then finally, and that will index the adaptive immunity. So basically, the different T cells will come there. T cell recognizes antigen, and then T cells also help in B cell to produce antibody. So this will happen within like 14 days of infection. But after that will probably clear the infection. But that is the important aspect of the adaptive immune response that I was telling you before, the memory. So after this virus is gone from the body, but we'll have these memory T cells or the memory T cells. So they will be there for years, like maybe 10, 20, 30 years, we still don't know, but they will be there in the body. What we'll do, like when next time when those virus or the bacteria enters into the body, they will act very quickly and very rapidly because they remember those pathogens. So that's how our body have the memory of like the past infection, and we can utilize this phenomena like to train our immune system for the vaccine. So like here, I'm just showing one of this curve. You can see, so this is the time of infection, and you have the pathogen in the body for weeks, like few weeks, 1 or 2 weeks, and then that will be cured, and there will be no pathogen in the body. But what you see here, like you see, this is the specific T cells, like their number will increase, like at the time of the infection, but like when the infection is not there, the number will decrease a lot. But there is some T cells will be still alive and they will be there in the body for years, like long time. Same thing for the B cells, and their number will increase a lot. And then after some time, their number will decrease slowly, but there will be a B cell which you'll remember those pathogens. And same thing for the specific antibody. You see the antibody level was so high during the infection, but the number will decrease over time, but still there will be some antibody available in the blood or the body, so they can react to the foreign pathogen. So this is the main principle. So what we want to do for the vaccine, basically, so you instead of using the actual pathogen, we can use some protein or some attenuated virus or the pathogen, so then we can mimic the infection and basically produce more memory B cell and T cell antibody before the actual pathogenic infection, and those will be in our body and they will be ready for the actual infection, so they can fight against those pathogens, which is very interesting, and I like this a lot. So lastly, like I want to say, like why antibodies are important. So this is the figure I just showing you. This is the virus membrane, and this is the spike protein, you see, and this is the S protein. Again, like we can see this actually best, that is because of the structural biology. And this is the host cell membrane. And what happens, like the spike protein will come and bind with the S2 protein. And then finally that leads to the fusion of the virus membrane and the host cell membrane. So the virus particle will enter the cell and release the RNA. But what, in most cases, what will an antibody do? Like I'm talking about this special kind of antibody, which is called neutralizing antibody. They will come here and bind to this place of the spike protein. So the spike protein will not have the epitope or the site to bind the S. Basically, they will block the binding, and so the virus will not enter into the body. So as many as we have this kind of antibody, which is actually beneficial for us. So at the same time, so they will not bind, and so with the spike, and basically there will be no fusion. So, but this is the way we are. Hello. This is the way we are thinking that we can make more of this kind of antibody or the specific T cells, so then we can block this we can block this infection. But in the same time, if you think about like what we are saying, like we had alpha variant, then beta, then gamma, delta, now omicron, and also we have now subvariant of omicron. So what is happening here? Like which is it's called evolutionary arms or natural selection or the evolution of the virus. Basically, we are keep producing. We have this our immune system which are becoming capable of like inhibiting those some of the virus. And like we had alpha first, and then we kind of become immune with the alpha, and we are having antibody everything for the alpha. So in the same time, virus is also mutating, and virus is also mutating its spike protein everything, so antibody can't bind. So that's how by chance one of these 10 might be become more like infectious, or they can spread easily like the beta. So that's how beta is generated. Like it has some mutation in the spike protein, so that can evade the immune response. Same way gamma, delta, or omicron, they started arriving. So this will be going on forever because we can't like eradicate all the pathogen. That will not be possible ever. But I think we can hope for coexistence because that's how nature works. So what are the different vaccine and what are the different vaccine strategy we can take? So in order to develop vaccine, we can develop many ways. So a vaccine can be a protein from the pathogen only. So like here, you see the spike protein. The spike protein can be a vaccine. If we can make in the same way as it is present in the virus, then I think it will be valuable. So we can just inject those protein into the cell or body, and our body will immediately recognize those as a foreign. As I told you before, like the MSC, T-cell, or antibody, so they will recognize those as foreign, and they will mount immune response. And we have these memory cells in our body, so they will recognize when the actual pathogen enters, like the COV2. And virus-like particles, because we have this protein, and then we can use those protein to make some particle, like nanoparticle or something, and then we can inject also. Another thing that we can use a DNA-based vaccine or RNA-based vaccine. Basically, instead of putting the protein, we have the DNA, which is actually codes for the spike protein or other protein, and then we can inject into the body. So the body will make, or the cells will, antigen-presenting cells will make this protein, and then we'll have immune response against those. Another aspect is like you take a different non-pathogenic virus, like non-replicating viral vector, and then you engineer some of the spike protein into those and inject those. Same thing for the replicating virus, like which is not pathogenic, but we inserted some of this gene from the pathogenic virus and then make this recombinant virus, which is not infectious to us, but it will have a protein and from the pathogenic virus, and then and our body can recognize those as a like foreign, and then we'll have the memory basal. And this is, and the last thing is the whole vaccine, which is the like the oldest way to make vaccine. Basically, you take the pathogen viruses like SARS-CoV-2, and you kill those virus, basically steril those, and then and so that it can replicate or create disease in the body, and then make those large quantity, and then use those as vaccine. Basically, inject those into the people. Um, Similarly, we can have like live attenuated. Those viruses are live, but they have limited capacity to create disease. So they will not be able to create severe disease. They will be clear off from the body very easily. So these are the different strategies uh, 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 like available for developing vaccine. And for the SARS-CoV-2, I think it was remarkable that scientists can develop these vaccines so fast. Uh, so, and also we have seen like a new technology that came up, which is mRNA. Um, and people are actually working with mRNA vaccine for a long time, but it was the time when it they actually worked for the first time. I'll talk about the mRNA vaccine later on, but um, like in India, we had Covishield, and we are also having the Covaxin. They are made from like the viral vector or the inactivated. I think Covaxin um, is actually inactivated virus, and Covishield is actually from the viral vector. So these are the two different strategies, but these are also effective and safe, and that can also protect us for the actual uh, virus infection. And I think there are other different strategies also going on, like making recombinant protein, the uh, spike protein. And these are the different companies who have been trying to develop those, or maybe the DNA molecule for the spike and then inject those. So this is a figure actually showing how one of these RNA vaccines looks like. Like it has the RNA, and RNA actually codes for the spike protein, and RNA is stabilized with some kind of lipid because it's negatively charged, and the RNA is also encapsulated with this phospholipid and this lipid mm, vesicle. So basically, the lipid will help this mRNA to enter into the cells, and antigen presenting cells, they will take those, and they will read this mRNA and a ribosome, which will make the protein from this mRNA, and antigen will be presented by MSC molecule, and also this protein will be expressed on the cell surface, and our immune system will recognize those as foreign and then start immune response immediately. Uh, so one thing I wanted to talk about, because this is a very powerful technology, because uh, now we have this mRNA. Now we can see like the different variants are coming up for the only for the SARS-CoV-2. So we can use this same vaccine very easily, because we just need to change the RNA sequence here. Like this was for suppose alpha variant, and then now we want to make for Omicron. We just need to change the sequence for the RNA, and that then the vaccine will be ready for the Omicron. And same thing, like we can have different RNA there. We don't have to, like, if you want to make vaccine, we need to go for like very different strategies, like to make sure the protein is stable, everything. But this is a very easy way to make different vaccines. So now, I think in the future we'll see many vaccines uh, for which are actually developed by this same strategy, like mRNA vaccine. And another important thing is that people are trying to develop this mRNA vaccine for the cancer, but that will take time. So another aspect I wanted to talk, this is for the student, because you see the mRNA was first identified, discovered 1960. And then it took a long time, almost 60 years, to develop the application of this mRNA vaccine or technology. It's like 2022. So there are many people worked on this mRNA, and also they worked on different aspects of it. So it's not only one scientist or two scientists. I think there is so many different labs and so many different PhD students, postdocs. They actually all work together for years, like decades, and this is more than 50 years. So, and finally, that actually helped to create one of those vaccines. So this is a continuous process, and here another important fact for this mRNA vaccine I would like to talk about is that, like, why is not possible, because, like, suppose we can easily make the RNA like long before, and then if we inject those RNA, they should make the protein inside the cell. But the problem is that our body, our immune system is also, they can sense this RNA from the outside the body. Like when they see this mRNA is outside, those can be a danger signal for the body. So body will start immune response against those. So so we don't need to have that. Like if you use this mRNA, the vaccine will not work efficiently. So we need to find a way to minimize the toxicity of this mRNA. So what was helpful is that one of this base is uridine in the mRNA is actually that's how our immune system recognizes those. But later on, scientists found out that if we use, instead of uridine, if you use methyl pseudouridine, which is like very similar structure but only difference in the base, and so we can evade the immune system, and there will be no toxicity that much, and also the mRNA will function the same way, basically go inside the cells and make the protein, and so the peptide and the MSC will be MSC molecule will present the peptide, and also the spikes protein will be also expressed on the cell surface. So this is a very small fact, but I think this is also very like we need to do research to find out like how we can manipulate the system so we can use the system for our own purpose. So so this is really a groundbreaking discovery. So conclusion. So what, and this is for the student, what I've like to show you that that research is important, mostly the basic research, and it's a long process, and many people, many students, many scientists are involved in this process, and it's not easy. It need to a lot of failure. It goes through a lot of failure, and finally you will be able to end up able to understand the process, and so that we can manipulate for vaccine development or drug and medical treatment. And sometimes people call about basic and application research, but if you think that everything is actually stand on the basic research, because that's how, when people found the mRNA, they never thought about that like mRNA could be used as drug. But in the same time, when like people started using this, started studying, and finally they realized that it has powerful. It can be a powerful way to design vaccine, drug, or medical treatment. So with this, I'd like to thank everyone for listening to my seminar, and I'll be happy to take any question that you have. Thank you very much.
Participants are requested, from that, if you have any question, you can ask, and students are also asked to question in Bengali version also. Actually,
yes.
তোমরা যদি কোনো কোশ্চেন থাকে তোমরা বাংলাতেও করতে পারো, কারণ স্যার আমাদের কাছে আমাদের এখানকার তোমরা মোস্ট পার্টিসিপেন্ট আমাদের ওয়েস্ট বেঙ্গলের বিভিন্ন কলেজ ইউনিভার্সিটি থেকে এসছে, তোমরা বাংলাতেও স্যারকে কোশ্চেন করতে পারো।
অবশ্যই।
তোমাদের বলছি, তোমাদের যদি কোনো কোশ্চেন থাকে, কোনো কোয়ারি থাকে, তাহলে তোমরা স্যারকে বলতে পারো, জিজ্ঞেস করতে পারো, স্যার তার আনসার সেটা বাংলাতেও তোমরা করতে পারো।
হ্যাঁ, আমি অ্যাকচুয়ালি এখন সিলেবাসটা জানি না, তখন তো আমাদের সময় বিএসসি তে অত ছিল না, কিন্তু
hello, Sunirmal Babu. I am Suman Pratia from
Yes, sir.
Kirkukar. I have little.
Oh, Suman.
I have one question related to the topic. May I? Hello, may I?
Yes, Suman. Yes, you are audible. You can have.
Yeah, yes. I am audible. Sir, first of all, it is a very illuminating lecture from your end. I have learned many things, but I have a little query. It is not a question actually. It is some kind of query from. I have read different journals and different popular articles and few of the popular articles from the reputed newspaper like Guardian or like New York's Time. But I have a question regarding that. Everyone is talking about the memory B cell, but no one is talking about the memory T cell. So, what is the probability of treatment of coronavirus through the memory T cell?
Thank you for asking this question. I think this is really a great question. Like we will not have memory B cell without without T cell. So immune response start with actually we need the T cell, and T cell will actually prime the B cells for the antibody production and having the memory B cell. So that's why in case of the AIDS, they will infect CD4 T cells, and that's why it happened like immunodeficiency disease. Like B cells are still there, but they are more prone to infection. So everything starts with T cells. So yeah, people usually cover B cell more, but because of the antibody. But actually, you need T cell first, and then B cell will come. So this is an excellent question. Yeah, thank you, sir, for asking that.
Sir, what I want to know exactly, not for T cell, means I want to know the memory T cell, the subunit, the subclass of T cell, memory T cell, and the cross reactivity of that T cell. Is it affecting to the coronavirus treatment or infection?
Yeah, cross reactivity can be affect it can affect because, but I feel like cross reactivity can help because there are many kind of coronavirus there, and we may have prior immunity for those T cells. And memory T cells, if you think about SARS-CoV-2, before, we may have some memory T cells which are cross-reactive for this SARS-CoV-2, but they may be originated from the past coronavirus infection. I think they might be helpful. Yeah, of course, they might be helpful because they can act more efficiently or will not take much time to act against us. And yeah, I think they can be helpful. Yeah.
Thank you, sir. Thank you.
আপনি মিউটটা খুলুন।
Subhan sir, many thanks. Sundar presentations, much more details, and very interesting.
Sir, I have a question. Sir. In our country, we have phase 2 type of vaccine, Covaxin and Covishin. Which one is more safe? Someone sir is there?
Sir, please unmute.
Okay, sorry. Yeah, I think Covaxin is actual whole virus attenuated or like killed. So it has all the different parts, like all the different proteins. And but the Covishield is only having the spike protein, and it's actually using a viral vector for that, and it's actually killed or attenuated. I think both of them are safe as soon as they are made in a like the actual they're following the guideline, everything. So that I think that's it. Same thing for the other vaccines too. Like there are always skeptical people about injecting those vaccines. Here also, like there are many people they didn't take the vaccine. They think it can be have some adverse effect to them. So, and also people are hesitant to give this vaccine to their children. But I feel like in India, like we always like to take the vaccine because of the MTB. I feel like MTB was really bad in past, so we always try to take the BCG, everything. And I think it's both of them are safe to take.
Sir, I want to add one point with your answer that the vaccine data provided by the both companies, by the Covax or Covishield, it is quite some actually 5% to 7% difference between the Covaxin and Covishield vaccine data, and Covishield is quite uh, actually 5 to 7% better than Covax in respect to the vaccine data what they have provided. That is my addition.
Okay, thank you. I didn't know that, but yeah, I think probably both are safe, but I think they might have little difference in efficacy. So some might work better than other.
Sumon, thank you so much. Shomita, Maitri. Shomita, please.
Thank you, Dr. Shobhan Roy. It was really an informative session, and I am very much sure that the precious knowledge you gave us will definitely help all the participants and particularly our student, and they enrich themselves. Thank you, Dr. Shobhan Roy, once again.
Thank you, all of you, Belda, Sumita, Madhusantati. I had really good time, and I'm very happy to share my presentation. And I also thank you, Professor Mahapatra.
Thank you, Shobhan sir. Thank you.
Yeah, it's a pleasure to see you again. And now I'll transfer to Mahapatra, and I think he'll talk about more about COVID, and I hope you'll get time. Thank you so much, and I think I need to leave because my son is already awake, so basically go. But thank you so much. Thank you. Okay.
Thank you.
Nirmal sir, please unmute.
Okay. So our next speaker is Dr. Pradeep. He is Chief Professor, Department of Microbiology and Director, AKB Environment Conservation Center, Raimati University. Dr. Pradeep did his PhD in the year 2008 from Vidyasagar University. He is now working as a associate professor in the Department of Microbiology, which I already told. Uh, he is also the director of Professor A.K. Bhutra Environment Conservation Center, Bangalore University. Dr. Das Mahapatra has significantly contributed in the field of microbial enzyme technology, environmental and industrial microbiology. He has worked on microbial nutrition and diversity, fermentation, probiotics, immunobiologization, bioinformatics, etc. He is presently working on metabolites. Dr. Das Mahapatra has published 3 books on microfermentation and enzyme technology, Chia Secret, Taylor and Francis Group, and another 2 on biofertilizers. More than 21 books chapter, 5 review papers, 6 articles, patents. He is also attended 8 training programs on microbial research, covered national and international conference, and delivered 24 invited lectures. Dr. Mahapatra has more than 21 years of research, 40 years and from UGC, Government of India, ICMR, Government of India, Biostatic India Limited, SEM Limited, and already awarded under its permission, and 6 are working. Dr. Mahapatra has received Junior Scientist in the year of 2008 and Environmentalist of the Year Award 2009 from National Environmental Science Academy, New Delhi. He is nominated as Fellow of the Society for Applied Biotechnology in the year 2011. He is also serving as an editorial board member of 3 journals and reviews of many journals of Elsevier, Blackwell, Springer, and Taylor and Francis. Dr. Mahapatra is also associated with many academic and research societies like FAB, France, NESA, New Delhi, VRSI, Trivandrum, AMI, New Delhi, PBVM, Calcutta, the Indian Science Congress Association, Kolkata, SAB, Dharwad, etc. So such type of research person we got for our international webinar. We are really proud, we feel proud. So Dr. Mahapatra, please.
Thank you, Dr. Giri. May I audible? Hello.
Yes, you are audible.
Yes, sir. Yes, sir.
Okay. So the patron of today's seminar, Dr. Manavendra Mondal, Honorable Principal of Belda College, the committee members, Dr. Madhuchanda Duari, Dr. Giri, Dr. Sumit Giri, and other all faculty members those who are associated to organize the seminar, my beloved students and other participants, research scholars. I am going to discuss about the benefits of probiotics with respect to the antiviral and some immunomodulatory effects related to virus with specific to COVID-19. So I am very much feel privileged as you are inviting me, and I am going to talk something related to probiotics. Basically, it is a very interesting topic, and we are now day by day encircled with many diseases, and our body systems is going to be ill day by day. So, what about the probiotics? If we think the in point of definition point of view, basically it is nothing but the live microorganisms and bacteria, most of them, and other organisms are also there. If they are present in an adequate number in our human body or intestine, then it gives many more health benefits. And whatever the microorganisms are concerned, we are divided into 2 groups. One is bad group of microbes and another are good groups. So the bifidobacteriums, lactobacilli, and some strains are E. coli. They are mostly helpful, and other many probiotics or helpful bacteria are there. As compared to other organisms, the Campylobacter, Clostridiums, and Enterococcus are most, and other things are there most bad bacteria. And the two or three things are very important. One is normal intestinal flora, and probiotic already I discussed. And in addition to that, the prebiotic. These are nothing but the non-digestible food ingredients, but which are required for the microbes, or they are acts as a food for the probiotic bacteria. So these prebiotic molecules helpful to develop the colonizations and more growth of the probiotic organisms. And another terminology is symbiotic. This is a combination of probiotic and prebiotic. And after that, the metabiotic, and it is a very new concept when the all the component or essential molecules of probiotics, they act as a direct beneficial effects on different diseases. We called it as a metabiotics. And these things are noticed in very early periods by the Eli Metchnikoff, and he noticed that the longevity of yogurt eating peasants, means the laborers in the year of early 20th century in Russia. So their age, their longevity, their skin glazeness, and other things are very high. So in that period, something is taken into consideration. So why these peoples are more healthy and more strengthened? What is the reason behind it? And if we think about a few words regarding the probiotic history, so the Eli Metchnikoff, first scientist who proposed the therapeutic use of lactobacillus or lactic acid bacteria. Then after that, their isolations and from the rancid milk by Lister in 1878, and one interesting features of the strain Sirota by the Dr. Mainur Sirota in 1930. So in this way, this probiotic science developed. In another way, if we think about the microflora of human body, you always find in everywhere there is a very good number of microflora, and they are encircling us in every day, every moment, every seconds of our life, and we are very much dependent on the microflora. And if you compare that, you can see the colon having the highest number 10 to the power 11 to 10 to the power 12 microbes as compared to the less number in stomach. So as and when the food items are digesting, so their populations also increasing. But as the number of microorganisms, number of bacteria, fungi, protozoa, viruses also are there. But how? What are the specialized characteristics meant to designate the probiotic organisms? So they are very simple. One thing is they must have to survive in the stomach, so they have to be acid and wild salt tolerant. And another property is that bacteria having the ability to adhere with the intestinal linings, and they definitely establish a temporary home or residence in the intestines. And in many ways, they must have the ability to give benefits to the human beings or animals like that. So they should have a leaf. They must be safe. They should not, or they must not exert any toxic effect. And other things like their pharmaceutical and topical applications they must have. So these are the main features for the microorganisms or bacteria to designate as a probiotic organisms. Another two interesting one is antibiotic susceptibility and antimicrobial activity. That means that bacteria must have the ability to synthesize some molecules that can kill the related pathogenic organisms. Again, this probiotic organism must be killed by any antibiotics. Otherwise, they become a resistant one. So antibiotic susceptibility and antimicrobial activity, two characteristics must they have, and many enzymatic activity also there. probiotics. So if these things are hostage in any microbes, any bacteria, then we called it as a probiotic organisms. So the World Health Organization, they have a specific guidelines how we can isolate and identify the microbes, and how we characterize them to designate as a probiotic organism. So there are criteria for selection, assessment, safety aspect assessment, and in that case the double blind placebo controlled randomized human studies are there to determine the efficacies, and their clinical studies. And if this organism or these strains are satisfy all these characteristics, we labeled as a probiotic. And there are all labelings like health beneficial effects, genus, species, strain designations, minimum number of viable bacteria, that thing must be there, and their shelf life, effective dose, and appropriate storage conditions. So after that, that product can be released into the markets. If you think about their types, then you can see the gram-positive bacteria, negative, yeasts, microalgae. So they also, these all the diversity of probiotic organisms, they give the health benefits to us. And just you see the slides here, you can see how or which many way this probiotic organisms helps to the human beings, starting from the mucosal membrane or accumulation in mucosal membrane, and reduction in the allergic levels, and so many way the synthesis of interleukins, and reductions of inflammatory activities. So in this way, they gives us many more benefits. So if we categorize them and their specific activity, then first one is it compete for adhesion site aggregations. So whenever the pathogenic organisms are there, so the pathogenic organism began replaced by the probiotic organisms due to their mutual, or we can say that their quick accumulations or utilization of the nutrients, and now nutrients became shortage to the pathogenic organisms. Now, in another way, the inhibitions of collagenase and reduction of inflammation-associated molecules, it enhances the host immune response and enhance the production of IgA and defensin. So these are the main basic principle behind it for immunological interactions. Then prevention of cytokine-induced apoptosis and influence on local and systemic immune response, or it provides the less or inflammations and tissue destructions. So these are the different way how they in different mechanism they gives us the benefits in our body systems. So there is a antagonism relationship with the pathogenic organism. In another way, if we see their application potential, so it is a very big list. You can see the slides. It is starting from the reduction of Helicobacter pylori infections. Basically, this Helicobacter pylori is responsible for ulcer, and it's a very long time it was a debated things. The peoples know due to the rich food, the ulcers are there, but now it was very clear by Warren and Marshall scientists. So this is due to Helicobacter pylori. So to reduce the Helicobacter pylori infections, the probiotic organisms are very helpful. Then the obesity, diabetes, and diarrhea, then enterocolitis, inflammatory bowel disease, so lactose intolerance, bacterial vaginosis, recurrent UTIs, and so many dental caries and cardiovascular risk, even the rheumatoid arthritis. So there is a application is a very high and very long list of application of probiotic bacteria. So you can see the many products are in the market regarding the probiotics and their which strains are there, and how their dose are mentioned, and it can be uh, readily available in market, so the people can easily consume. So in a different way, these probiotic products are there. like the Yakult, and then different company they gives the name in a different way, but everywhere the microbes are there. So one thing is in common man, for common man, this the dairy-based probiotic food is very helpful, like the drinkable fresh milk and fermented milks, yogurt, cheese. So if we make a habit in our life, so it is very helpful to us. And non-dairy based probiotic products, or at present there is so many strain specific probiotics are available to treat a specific disease. So if it is think about the non-dairy based probiotic product like vegetable based, fruit based, cereal based, and meat based probiotic product. So these are nothing but the fermentative products. So vegetable, fruit, cereal, meats are now fermenting by specialized strains of microbes, helpful microbes, good microbes, or probiotic microbes, and their effects gives the health benefits. So there is a big list how these products are coming up in the market, and research are going on in the sector of food microbiology and fermentations sector. So you can see the different fruits, vegetable, soy-based products, cereal-based products, and non-dairy products. These all things are there. So there is a literature. You can see everything and how these fermentative products are there or probiotic products are there. And this is well known to all of you regarding this probiotic product or fermentating product. So these things are related to human being, but the another way, the aquaculture sectors also is now dependent on probiotics due to the toxic effects of the other chemicals and antibiotics. So the governments banned, and other foreign countries the banned the utilization of antibiotics in the aquaculture sector. Again, in another way, the bioremediation is also going on through this probiotic organism. So the application of probiotics in human health, the application of probiotic in aquaculture, in other animal husbandry, these all things are now coming to dependency on the probiotic organism. So the quality of water improved, and disease may be controlled, growth promoters and nutrients and enzymatic contributions to digestion of the molecules and improvement of immune response. So this is also some literature. The strains are targeted, which are the probiotic species, and target aquatic species like water quality, control disease, growth promoter. And basically, this is available in the literature. The interested person can see. But thing is that the environment may be cleaned, and the helpful microbial populations must be increased in the society. In another way, the colorectal cancer, in that case, we got a recent project on cervical cancer, and this is related to applications of bacteriocyanogenic lactobacillus to treat the cervical cancers, or again it related to also colorectal cancers. So the intestines, or you can see the stomach, colon, these everywhere the microbes are there, and how we consider or improve the populations of probiotic organisms in every sphere of life. This is a challenging task. Now we are going to discuss about the human probiotics. So you can see I am just talk about the strain specific. So the microbes, or the people can say the dairy product probiotic is sufficient to combat all the things. No, the answer is no, because it is required a strain specific, and it continue for the living states. When you consume the antibiotics, then your all microbial flora become disturbed. So probiotic flora also disturbed, killed. So it requires a specific strain of microbes, and that microbe must be sustained in the intestine or any other areas. So there is the gastrointestinal probiotics, skin probiotics, scalp probiotics, and you are very aware about that. The scalp is a very dangerous disease, a fungal disease. It cannot be curable. But scalps or strain-specific scale probiotics are now coming into the market, so you can use the scale probiotics. Probiotics of the oral cavity, probiotics of the underarm and feet, and urogenital probiotics. So what are the, which strains are more or the more important players? Basically, Lactobacillus plantarum, Bifidobacterium infantis, Lactobacillus salivarius, Lactobacillus bulgaricus, KG, brevis, and Thermophilus. They are the most players or the big players in the probiotic market. So basically, the Lactobacillus acidophilus, this produces the lactosidins and acidophilin. These two molecules are very much helpful to kill the pathogenic organisms like Salmonella, E. coli, Staphylococcus, like that. And you can see the Lactobacillus acidophilus. After that, Bifidobacterium bifidums. So this is absorbed large quantities of ferrous ions and inhibiting the growth of bad bacteria. And these bifidobacteriums basically reside in the mucous membrane of large intestines and vaginal tract, and it produces most beneficial forms of lactic acid and acetic acids. So this is the structure of Bifidobacterium bifidum. So as the name is there, you can see the structure of the bacterium is somehow different. Then Lactobacillus rhamnosus, it basically relieve the hypersensitivity reactions and intestinal inflammations in individuals with eczema and food allergy. So that organism is helpful. Lactobacillus plantarums, it also produces the lactoline and natural antibiotic and synthesize amino acids lysines, which is antiviral molecules. Then Bifidobacterium infantis, it present in the intestine of infants and adults. So this is again, it gives a health beneficial effect. Lactobacillus salvaricus and Bulgaris, it is a very well-known microbes, and it provides the uh, fermentations of natural yogurts and provides the huge amount of lactic acids. Lactobacillus kg, and it secretes the peptidoglycans, a recognized substance that supports the body's natural defense. Lactobacillus brevis synthesize vitamin D and K in addition to lactic acid. Then Streptococcus thermophilus, so it breaks down the milk products by producing lactase, and it helps the individuals that are lactose intolerant. And many pathogenic organisms are destroyed by these organisms. Now come to their actions. Basically, these probiotics have the antimicrobial activity by decreasing luminal pH, secrete antimicrobial peptides, inhibit bacterial invasions, and block the bacterial adhesion to epithelial cells. Another part is enhance the barrier functions, like increase the mucus productions. So it is a very defensive systems, and in immunomodulations, the effects on epithelial cells, dendritic cells, monocytes, and macrophages. There is a effect on lymphocytes. So you can see their mechanisms. So basically, the adherence and subsequent stimulation of gut-associated lymphoid organs and upregulation of mucin genes takes place and enhance secretory IgA and maintain normal macrophage functions. So it is a competitions for essential nutrients, and you can see in picture how it creates. So here the production of antimicrobial substances are there, and by which the pathogenic organism populations are destroyed. So this is a principle of destruction of toxin receptors, and complete term exclusion principles are there, and organic acid productions and enhance the barrier functions or the IgA secretions. So you can see the intestinal microbial populations are there, and if the probiotic, the yellow colors are probiotic organisms, if their populations are increasing, then you can see the mechanism of activation of antigen presenting cells, and finally the interleukins are synthesized, and it synthesize the actions like anti-tumor or antiviral activities and reductions of the allergic response. This is the same thing, but I come to the mechanism here. Here you can see the quorum sensing activities. The pathogenic organisms, they having the activities like the quorum sensing systems. So if they are present in a threshold levels, then only they secrete the toxins. So before that, when they are very small in number, they cannot synthesize the toxins. So this activity also hampered by the probiotic organisms. It gives a much more benefit to the human beings. And here you see one by one the mechanism of actions. So one thing is competition for nutrients and prebiotics. So as and when the pathogenic organism populations are increasing, or in that case the probiotic organism consume much more the molecules or the prebiotic molecules, and their populations are in high in numbers. So these things are happening. And another way, the bioconversions principles are there. Then production of growth substances and vitamins are there, and in another way, you can see the direct antagonisms are there, so pathogens are destroyed in the body systems. Then the barrier functions are there. Then enhanced innate immunities by activations of dendritic cells. So these are the main mechanism by which uh, it can be gives a health benefits to the living systems or human body or animals or anything. So the probiotic have a immunomodulatory effect, and the bacteria have the ability to interact with the epithelial and dendritic cells, so by which it creates a innate immune system response. The common structures called pathogen associated molecular patterns shared by the vast majority of the pathogens. These are the basic principle behind that. And we just discussed about the immunomodulations. Now come to the lactose intolerance. One thing is that many people are not able to consume the lactose because they don't have the synthesis of beta galactosidase. Or in that case, if the probiotic organisms are there, so the beta galactosides are synthesized by the probiotic organisms, and it can easily digest the lactose, or due to their fermentation activities. Now the yogurt or the yogurt bacteria hydrolyze the lactose, so it converted to glucose and galactose. It is absorbed by the body. So in this way, the lactose intolerance peoples, they can easily consume the milk product. And another thing is antibiotic associated diarrhea. So basically nowadays, whatever the disease are there, but the doctors are prescribing the antibiotics, or the peoples are interested to consume the antibiotics from the medicine shops. So it creates the problem in the human body. So if unknowingly the infection is not by the bacteria or infectious disease controlled by the antibiotics, so what happens in that case? So people consume the antibiotics, so it is disturbance in the intestinal microbiota, and as and when there is a dysbiosis in the intestines, the pathogenic organism, including the clostridiums, they become overgrow and they produces the toxins. So the probiotic organism in that case can balance it, and it creates a microbial balance in the intestines, so it reduces the antibiotic associated diarrhea. Now come to the rotavirus diarrhea. The same things is in case of rotavirus also. So it is a very common and requires hospitalization 2 to 3 days. But oral rehydrations and probiotics like lactobacillus, bifidobacteriums, so they can reduce the hospitalization process, or the patients can cure at home, or maybe it requires 1 day hospitalizations. So the non-lactic acid bacterial probiotics and disease-specific probiotics, or the strain-specific probiotic, function-specific probiotics are there, and probiotics for specific subpopulations like the infant, elder populations. So these are the different specific organisms are there. So, in one thing is that if the science of probiotics hold up in the clinical trial, yogurt could replace many pharmaceutical drugs, but the strain must be alive in the human body, because the people think once I consume the probiotics is always help me. But inside our body, whether they are surviving there or not, this is a big question. So we don't know whether they are alive in the body systems or they are dying. So now I come to the coronavirus. One thing is about 200 different viruses can cause the common cold. It is a very general thing, but in 2000, 50% caused by the rhinoviruses, and only 15 to 20% caused by the coronaviruses. And that thing is was earlier also and at present day also, but some infectious strains are there nowadays. And this ACE2 or angiotensin converting enzyme 2 expressed in the intestinal epithelium regulates the ecology of the gut microbiome, so intestinal amino acid homeostasis. And these ACE2 receptors are markedly downregulated by the entry of SARS-CoV-2 into cells through membrane fusions. So this is the basic problems, and the ACE2 can consequently lead to an altered microbiota that confers susceptibility to the inflammation of the gut. So I am discussing in one way, and so many other factors are there. So what is the relation of virus entry and dysbiosis in microbial community in the human system? That things are I am discussing, and so many other things are there. I am not going to that. So as a when this virus entered into human body, and whatever the dependency of human body towards the microbes, they became destroyed or imbalanced. Now, as they imbalanced, so pathogenic organism populations are increasing. So this is a dual effect, or whatever the treatment related to lungs failure and other diseases, like the consequences after COVID, this is mucormycosis and other diseases are there. This is due to the imbalance of the microbial flora of human body. So once they destroy, instead of they providing the health or the immunity or whatever these things or good things are there, they are destroyed. In another way, pathogenic organism populations are increasing. So secondary infections is a very high. So it resulted in the lungs failure and other things are there. So given this evidence, the bacteriotherapy could represent a complementary resource for the prevention and restoration of SARS-CoV-2 intestinal mucosa damage through the modulation of gut microbiota and decreasing related to inflammation. So, first thing is we have to take care as and when the virus infections, not only the coronaviruses, any viruses, when entered into the human body, so human systems or every people should take care their intestinal microflora, because they gives the a 27 into 24 into 7 support to the human beings. No other medicine, No other nutrients can give that type of support. The patients with COVID-19 have an altered gut microbiome with depletion of beneficial commensal and increase of pathogenic organisms. There are specific organisms, whatever the research outputs are there, the organisms like Eubacterium, Ventriciums, the new commensals are there, then Faecalibacteriums and Roseberia. So these are commensals. So they increasing, and pathogens like Clostridium, Actinomyces, bacteroids, or whatever the mucormycosis their populations are in. The lungs have their own microbiota. Maximum death that is due to the lungs failure. So if patients come to the ventilations, and when they are in ventilations and 100% risk are there. So there is the factors. So one thing is lungs have their own microbiota. And gut-lung connections has been described wherever by host microbes, and microbe and immuno interactions can influence the course of respiratory diseases. So when the lungs become failure, that means there is a immunological response as well as gut-lung and microbes interactions are there. So the gut microbiome has a critical impact on systemic immune response and immune response at distant mucosal sites and including the lungs. So not only we think earlier that the microbes present in the gut. So whatever the relationship, whatever the interaction, whatever the modulations, so these are related to restricted in the gut. No. So there is a gut-brain axis, gut-lungs axis. So their molecular interactions are there. They are governing the alterations and risk management. So all things are going on. So the probiotic strains improve level of type 1 interference, increase the number and activity of antigen presenting cells, natural killer cell, T cells, as well as the level of systemic and mucosal specific antibodies in the lungs. So if we take care about the gut or about the intestines, then automatically other things are taken into care. So there is also so many evidence that probiotics tends modify the dynamic balance between pro-inflammatory and immunoregulatory cytokines. So here is the questions, and that allows the viral clearance while minimizing the immune response mediated damage to the lungs. And this might be particularly relevant to the prevent ARDS, a major complication in COVID-19. ARDS means acute respiratory distress syndromes. So, the people are going to the hospital or their health insurance, all are related to the disease. But nobody take care about their insurance or the taking care of their health. So if we think about the taking care of health, then it is very easy to combat any pandemic or to combat any type of devastations in the health systems. And many documents are there, many research points are there. I am not taking details on that, but use of recognized prebiotics, that means the galactans and fructans, to enhance the propagation of probiotics and indigenous beneficial microbes should be recommended as part of the overall strategy to flatten the curve of viral infection, not only COVID-19, to flatten the viral infections. And the anti-influenza, some records are there. I am not going to the details. So the probiotics became part of the armamentariums to reduce the burden and severity of COVID-19 and add enough oxygen and enough fluid to your body daily, so the ventilators could be avoided if you would be infected. So the gut lungs axis and gut brain axis and gut microbial interaction should be maintained in every seconds of our life. Then only it gives us healthy. and this is not the new things. So in 400 BC, Hippocrates said death sites in the bowels. A bad digestion is the root of all evil. And there are some of the publications related to the probiotics, and I am thankful all the collaborators as well as the funding agencies those who are give funds, and recently not added here the ICMR gives a project on probiotics also. Myself and our senior Professor Subhash Chandra Jena, uh, now working on that probiotic and cervical cancer. So, thanks to Keshav sir and Partho, the Behra sirs, and the earlier students and present students also working on that. And thank you. This is our my present university gate. And after all, many thanks to the students for their patience, the teachers, and the honorable principal of Belda College, Manavendra Mondal sir, for organizing this type of seminar. Now it is open for discussion.
Thank you, sir. Thank you, Professor Dr. Pradeep Kumar Das Mahapatra. Thank you, sir. At first, good evening, everybody. Dr. Pradeep Kumar Das Mahapatra has discussed a newly aspect of COVID-19, that is the health benefit, antiviral, and immunomodulatory effect of probiotics to combat COVID-19 pandemic and co-infection, and also its future perspectives in a very nice and simple way. We are really enlightened with your presence, knowledge, and informative speech. Thank you, sir, again. Thank you.
Thank you, madam, for your appreciations. So, any question from students? Okay, if there is no question, so thanks. Thank you.
Dr. Sunil Malgiri present. Dr. Sunil Malgiri
Present. Yes, I am present. Yes, Manisha. Now the session is open for discussion. If you have any question, then you can ask Dr. Pradeep Dasmapatra. Students or participants, if you have any question, you can ask Dr. Pradeep Dasmapatra. Dr. Pradeep, I have a question.
Okay, sir. Please.
So, which one is safe? Taking of probiotics or taking of vaccine?
No, two things are different. Two things are different. Vaccine gives you the temporal immunity for 3 months, 6 months, like that. So if the infection is high in the surroundings, so it is always a necessity for taking the vaccines. But the vaccine gives the protection to a particular virus, and probiotics gives the protection from any kind of virus, any kind of pathogenic attack by increasing your body systems. So one thing is for immediate actions, and another thing is for continuous activity. So both are required.
Thank you, Pradeep.
থ্যাংক ইউ স্যার। আর আমাদের মধুসন্ধ্যা দি কোথায়?
আচ্ছা স্যার,
ম্যাডাম, এনি কোশ্চেন?
I have a question, sir. Any food which protect ourselves from COVID-19 virus? There is any food? There is any food which protect ourselves from COVID virus infection?
Okay, there are so many foods, but I am discussed today only the probiotics, and the probiotics, strain specific probiotics, not only a common probiotics, but food items are there which are basically immunostimulating, like the natural products without any toxic molecules. If we consume day by day, so it gives us the immense benefit. But in addition to that, the psychological thinking, it is also important. I am feeling well, so every person's to consider their choiceable food, fresh food with the fresh mind. So it in addition to the probiotics and the prebiotic molecules, so not only the COVID, any viral infection can be prevented.
Thank you, sir.
Okay. If there is no more question, so if organizer permit me, so I can be conclude here for today.
Dr. Giri, where are you?
সুনির্মলা মিউট হয়ে আছে।
Monisha Das. So on behalf of Monisha Das, I want to thank for Dr. Pradeep Das Mahapatra, our second speaker, a second research person from Raiganj University Department of Microbiology, for his nice presentation and nice informative presentation for us. Students also enlightened themselves from the topics or from this webinar. So thank you. Now I invite,
sir. Thank you.
So now I invite Sumit Giri, our Joint Secretary, Departmental Head, Department of Zoology, Belda College. Sumit is there. Sumit. Sumit is there. We have come to the end point, valedictory section. So Sumit, where is Sumit? I think there is also a network problem of Sumit Giri. Sumit, if we have connection, please. Sumit, so I think there is network problem of Sumit Giri, and on behalf of Sumit Giri, I want to put up thanks all the participants, and I want to say that good evening everybody, as all good things comes to end in life. So is the webinar also. On behalf of the organizing committee, myself Dr. Sunirmal Giri, convener of this international webinar, take this great opportunity to put thanks directly on immunological aspects of COVID-19. At the outset, I express my heartfelt gratitude to our resource persons like Dr. Shobhan Rai, staff scientist, Department of Biochemistry and Molecular Biology, University of Chicago, USA. And another resource persons, Dr. Pradip Kumar Das Mohapatro, Associate Professor and ex-head Department of Microbiology, Director, PAKV, Environmental Conservation Center, Director, Additional in charge, Business Incubation and Innovation Center, Raigon University. We are already enlightened with their knowledge and presence. Thank you, sir. We are thankful to our Honorable Principal and Chief Patron of this webinar, Dr. Manovendra Mondal, for his enthusiastic support, and without his moral, our heartfelt thanks to faculty members of different universities, colleges, and research scholars. Colleagues, Dr. Sumit Giri, HOD, Department of Zoology, and the Joint Secretary of this international webinar. Dr. Madhusundar Diwari, Srimati Monisha Das, and Sumita Maiti, Faculty, Department of Zoology, for their moral support and active participation to make this webinar a great success. Our special thanks to Mr. Subir Shah, don't mind Subir. Subir Shah, Faculty, Department of BCA and Computer Science, Belda College, for his technical support. Without his support, this webinar cannot be a great success. With these warm words and a kind of message, we move to the end of this webinar, international webinar. Thank you all. Have a nice day. Thank you. So, and lastly, I want to say that all the participants who participate in this webinar, each fill up the feedback form, which is already given to our chat box. It is not provided in the our WhatsApp group. Only it's available in the chat box, and it is active only for this midnight. So, for certificate, it is mandatory to the so in our thank you, and to you also. So I request all the participants left this webinar after filling up the feedback form. And within 1 week, we will send your certificate.
Okay, Sunil sir.
Okay, Sunil, ভালো থেকো।
আচ্ছা, আমি তাহলে লেফট করছি।
Yes.
Thank you to all the faculty members of